Dylan introduce our speaker for today.
Okay, thank you, Danielle. So it is a great honor and pleasure for me to introduce today's Grand Round speaker, Dr. Franck Barrat, who received his PhD in 1998 in Paris, France, and then did a postdoc there and came to the United States in 1998 to conduct a postdoc fellowship at DNAX research in Palo Alto. He then, after his postdoc, moved in 2001 to DNAX, fairly close by there in the Bay Area, and stayed with that company for a good number of years, climbing up in title and responsibility. Ended up as a senior research fellow there at Dynavax until 2012, where he was recruited to Hoffman & Roche in Nutley, New Jersey. And I suspect strongly it was Jacques Bangerot who recruited you over there. Jacques was there for a good number of years. Then in 2013, Dr. Barrat moved over to the Hospital for Special Surgery, where he has been ever since, becoming a professor in Cornell at the same time in 2014, as well as in microbiology and immunology at Cornell. And since 2016, he has an endowed chair at the Hospital for Special Surgery. So Franck has published a great number of papers, lots of important initial observations. Much of his work has focused on plasmacytoid dendritic cells, the role of interferons in not only lupus, but also in scleroderma. And I overlapped with Dr. Barrat an NIH study section. He has served in many of them since 2007. And we overlapped in the hypersensitivity, autoimmune, and immune-mediated diseases study section, where he served from 2019 to 2023, and I think my years were about the same. So I am really pleased that he's here to talk to us about plasmacytoid dendritic cells, which clearly are important in autoimmune diseases. And so the microphone is yours, Franck.
Thanks, Thomas. Thanks for the invitation. And also thanks for this time in the study section. I always say that you learn a lot about people in study sections, about their knowledge and fairness and how they approach research. So I think I've learned a lot from you. So it's great to see you again.
That's okay. Why you are here, exactly.
All right. So today we're going to talk about pDCs. So I'm going to introduce some stories about how they can contribute to fibrosis, systemic sclerosis in particular, and also how they get also impacted themselves by the microenvironment, the tissue microenvironment, and the high stiffness. So there's kind of two parts into that talk, and the one when I'm going to be focusing first, it has been a very strong collaboration with the group of Jean-Charles Guéry, in particular Bérénice Fass-Lopez in Toulouse, in France. They have experts in X chromosome inactivation, and they've done some of these unique experiments. Right, so this is just my disclosures. So yeah, type 1 interferon is probably one of the most robust and prevalent pattern of pathological gene expression in body and tissue in multiple autoimmune disease. And the field, I mean, the role of type 1 interferon, in particular lupus, had been suggested back in the 1970s, but it's really only with the emergence of new technology and microarray that groups like Virginia Pasquale and Tim Behrens and also Peggy Crow really identify what's been now used as this interferon signature, basically a set of genes which are chronically expressed in patients, sometimes for years, and that correlate more or less with disease activity. I think there's been much more refinements on this. And this kind of discovery opened the field in the early 2000s. These papers came out 2003, 2004. And I have to say, it's quite remarkable that less than 20 years later, we had the approval by the FDA of the first drug targeting type 1 interferon receptor for lupus. So in less than 20 years, we went from this early discovery to an actual drug that's being used today in lupus. But obviously, there's a lot of questions. What are the pathways driving type 1 interferon? What are the cell types involved? Other disease where this kind of approaches could be used for the benefit of patients. And I think the role of nucleic acids, but also of cells, such like plasmacytoid dendritic cells or other cells, obviously, to the contribution is kind of still an ongoing discussion and an ongoing debate. The pDCs produce very large amount of type 1 interferon, but they're very dependent on the toll-like receptors, TLR7, TLR9, and in some cases TLR8. While other cells like myeloid cells, other non-immune cells are going to produce type 1 interferon, but using sometimes TLRs, but also some of these other sensors of nucleic acids. And the question has been, what is the relative contribution of these cells, whether they contribute to different type of disease or maybe to different type of tissue involvement. But clearly, the role of nucleic acid sensing is quite important. And one of the reasons why it's such an important part of the immune responses in general is because of the redundancy of that system to sense nucleic acids, which basically you cannot change nucleic acids. And so you find receptors like the toll-like receptors, which are into the endosomes that sense double-stranded RNA, single-stranded RNA, double-stranded DNA through TLR3, 7, 8, or 9. But you also have CGAS, which are cytosolic, the RIGAs, MDF5, also M2. And these different sensors that sense different type of nucleic acids are located in different organelles in the cells, but they also associate with different adapters. The TLRs use MID88, except TLR3 that's using TRIF. CGAS is using STING, which is associated with the endoplasmic reticulum, while the RIGA MDF5 use MAV, which is with the mitochondria. And all these start to trigger NF-κB, TBK1, and the ARFs are going to drive type 1 interferon. So again, the question is really, how is that articulated during an immune response, but also potentially during autoimmunity? So Marco Colonna had this nice diagram that he published years. I've used that slide over and over, where he basically tried to put like a temporal response to antivirals and the presence of type 1 interferon, with the idea that plasmacytoid disease come very early on. They depend on the TLRs. There's very few of those. It's less than 1% of the PBMCs are pDCs. And they make this kind of early wave of type 1 interferon. And then you have basically involvement of many other cell types using TLRs, the RIGI, DNA sensors, and all these contribute to type 1 interferon. And when I look at that slide, it always strikes me that maybe this is what happened also during an autoimmune response. I mean, at the end, an autoimmune response is an immune response. So although it's directed to autoantigens. So when it comes to pDCs, I'm just going to show a couple of slides about these cells. The first one is, this is a horrible name because pDCs are not dendritic cells when they are in circulation. These cells have this kind of a round, it's almost like a very large T cells. They don't have any dendrites. They can't present really antigen as such. And the cells originate from the bone marrow. They circulate into the blood. There's basically absent in tissue in steady states, but they get recruited very, very quickly after any type of injury. And when they get activated, then by nucleic acid, TLR7 or TLR9, they really generate this astronomical type 1 interferon response. If you get primary pDCs from the blood, stimulate them with 7 and 9, and you look after 8-10 hours of stimulation, basically 60% of every genes that's being induced are type 1 or type 2 interferon. And if you add the interferon inducible gene, is pretty much 80 to 85% of every genes being produced is type 1 interferon response. And indeed, these cells have very low antigen presentation capability. They don't express CD86, CD80, CD40 very low, and they kind of really like interferon producing cells. That's what they do. They scream type 1 interferon. But then if you wait a little bit longer, and you look at these cells 24 hours or 48 hours later, that's how they look like. Now they actually look like dendritic cells. They start expressing co-stimulatory molecules. They can't make much interferon at this point. The entire machinery has been shut down, and now they can contribute to more like antigen presentation, especially through the class 1 pathway. So this kind of dual function of these cells has led to this very kind of pDC-centric view of the immune system, which is that through production of interferon, but also through presentation, these cells can contribute to both innate response, but also to kind of the adaptive responses. So the way I've often described these cells is these cells are very adjuvant cells. They basically get triggered in tissues through any type of injury, and then they secrete all these type of interferon and start influencing other cells, NK cells, T cells, B cells, and they basically start immune responses. And in my previous company that Thomas mentioned, at Dynavax, they have developed a vaccine using a CpG, which is a strong agonist for TLR9, and just basically use the same surface antigen of hepatitis B. And now they have an approved vaccine called Heplisav that shows very strong adjuvant effect of just triggering TLR9 signaling in pDCs. So that shows some of the data, and CpGs have been put in many different vaccines over the years. So it really stresses the idea that this kind of adjuvant properties of pDCs. So what about autoimmunity? So a role of the kind of this TLR pDC axis has been well described in many autoimmune situations, in lupus, Sjögren, arthritis, systemic sclerosis, and others. First, you see these cells trafficking. It's very well observed into the skin, these lesions of CLE in patients. But you find pDCs into the glomeruli of SLE patients. You find them into the joints of RA patients. So they really like to go and sit in this tissue as long as there's some kind of an insult or injury or lesions. And then the respective contribution of pDCs across all these different organs and in circulation was tested in 2 separate trials, which I'm going to summarize in one slide. So two trials have been trying to target pDCs in lupus patients. And one was to use the anti-BDCA2 antibody. It's not an entirely super specific molecule, and also it does not deplete the pDCs, but it delivers this kind of inhibitory signal. So although the cells are not necessarily depleted, they still can't make much type 1 interferon. The other approach was using the anti-ILT7 antibody, which is more of a depleting antibody. The results in both trials were quite comparable. So in this first phase 1, phase 1b, 4 placebo, 8 patients being treated with the drug. And then what you can see, it might be a little small, I'm not sure. But basically, if you look at the interferon signature in the blood, it goes down at 24 hours. But also by week 1, week 2, it's basically back to normal or abnormal, suggesting that pDCs probably play some role in the presence of the interferon signature in the blood. It might not be as critical in terms of long-term presence. However, in the skin, the data were much more striking. Looking in histology, this is a staining for MX1, no, for IFMT3. You see basically, even at week 4, the complete abrogation of the presence of type 1 interferon in the skin. And this led to interesting clinical observation with reduction of lesions score into patients which are treated, which are the ones which are in green, even at week 12. Only two patients did not respond to treatment. This was the patient that had discoid lupus, which could be just these 2 patients did not respond, or discoid lupus might not be as pDC dependent. The other approach was very similar, with the classes going down, inhibition into the skin also in patients. So it's been published a larger study by Victoria Reth and others in New England Journal now 2-3 years ago with a larger population, and I think this is an approach that's going. But I'm using this just as an illustration that maybe pDCs may have more of a dominant role in the skin, but maybe not as kind of the overall response in terms of type 1 interferon. Okay, so what about systemic sclerosis? So systemic sclerosis or scleroderma is really the prototypic multi-organ fibrotic disease. It's very heterogeneous. Paul Klee is probably one of the most famous patients. This is one of his last painting when he was very suffering pretty hard from scleroderma that he called Death and Fire. And that disease really kind of combined three things at the same time. There's fibrosis, there's vasculopathy, and there's also autoimmunity. So clearly there's an inflammatory environment. There's a very strong female predominance, limited diffuse cutaneous lupus, different forms, whether you get involvement of internal organs, but there's very little treatment which are offered for this patient. Steroids usually are not used because they have toxicity, and they're very still high morbidity and mortality in these patients. And this is some of the expression of the disease. So the renal phenomenon, the vasculopathy, but also the skin thickening and change of pigmentations and involvements in the lungs with this interstitial lung disease. So what we're going to talk about is a lot about, what about these pDCs? What is special about them and how they can contribute to fibrosis? But also I'll touch on some of the questions about what happened then to these cells when they get into the organs and how they get impacted by the tissue microenvironment. So one of the initial study that suggested that pDCs may actually have an important role was a paper by Team Ratstacker that published in New England Journal in 2014, that these cells had a chronically activated phenotype and they were secreting this excessive amount of CXCL4, which is platelet factor 4, mostly known as being produced by platelets. But here that it was showing that you just put the cells, the pDCs in culture, don't do anything, and they start secreting CXCL4. SHARE1 analysis also confirmed that there was a slight increase, not as much as what has been shown by Team Ratstacker, but still some excess of CXCR4 in patients. So we started to look at this, and the first observation is by looking at biopsies, skin biopsies. We could identify the presence of pDCs into the skin of a patient with scleroderma, which was associated with a reduction of their number in the blood, which is a classic sign of activation of pDCs. They basically get away from the blood to go to the tissue. We confirmed that they indeed produce a little bit of CXCR4 in a spontaneous way, and in some way also CXCR4. And I'm going to go back to CXCR4, but there's quite a lot of literature now that has come up on how this chemokine can regulate pDCs, but also macrophage and B cell functions, a bunch of papers if you're interested. So in order to see if they had kind of a causal, are they just attracted to the skin, or do they actually contribute to disease? We reported a few years ago that if you use the BDCA2-DTR mice, which allows you with DTR toxin to just remove pDCs in mice. So if we do the bleomycin models of skin fibrosis that leads to skin thickness, when we were depleting the pDCs, we observed quite a pretty strong phenotype with, so here we've depleted for the entire time of the experiments, and we see reduction of skin thickness. We quantified collagen that was also reduced in the PDC depleted mice. Reduction of some of the profibrotic markers, alpha SMA or TGF-beta, and obviously as almost a positive readout, all the interferon genes were decreased into these mice. Another study was done by Francesco Delgado. It was reported using skin node mice that were xenotransplanted with human cells, and again, when treated with NDBCA2, it could prevent fibrosis into the bleomycin. All right, so if they can actually have a causal role, what do we know about the cells in patients? So in order to approach this, we wanted to do a single cell RNA-seq approach. But this is a little tricky when you work with cells which are less than 1%, or sometimes, especially in patients, they basically less than 0.1, 0.2% of the cells of the total BBMCs. So there's been some studies that have been done to look at this. There's this pretty massive work that was published 3 years ago of very comprehensive single-cell analysis in systemic sclerosis using both skin samples and the blood from almost 100 patients and 56 healthy. And they did a pretty remarkable job in terms of annotating and identifying different cell subsets. And they indeed found that in the skin of patients, they could see the presence of pDCs, slightly correlated with the magnitude of the clinical scoring in these patients, but obviously no pDCs were observed in healthy skin. Virginia Pasquale published an approach using in SLE patients. She had also pretty large cohorts looking at, over 275,000 PBMCs to look at different subclustering of pDCs in patients. And there's been other studies. There's been more studies. I'm sorry if I don't quote one of yours, but there's been quite a few people trying to do this kind of single cell work in SSE or SLE patients. But I think they all suffered from the same issue. It was the number of cells that you could analyze. In the SLE study, you get 655 pDCs. In the very large science work, there was like 3,800. And in the one I just mentioned, this massive study, they basically had less than 500 pDCs into their data sets. So we felt we have to kind of trick the system a little bit. And we designed a strategy where we started with 4 healthy donors, 4 patients, all females, age matched. And before we ran the single cell, we first depleted the T cells and then enriched the pDCs and then mixed them back at a 1:2 ratio. And in that case, it allowed us with a sample of 8 subjects to get about 10,000 pDCs. So obviously this has some caveats. One of them, there's a price to pay. You lose the T cells, but also we having only a representation of 4 patients. Well, when you do these larger studies, you have a better representation. But this being said, we thought we would be able to maybe get some interesting observation in some of the different kind of subset present into these patients. So when we did the cluster analysis, we observed 5 subclusters of pDCs, and clearly we could see that there were some of those that were overrepresented into the patients as compared to healthy donors, and this was the cluster 0 and the cluster 2, while others were overrepresented into the healthy donors. So what do we know about these clusters? With no surprise, we found that these are the clusters which express the highest levels of type 1 interferon genes. So they carry the interferon signatures. It is basically enrichment of a population of pDCs that have interferon activated phenotype, which kind of match with looking at kind of the maturation of these cells into the patients. But then the question became to us, so how do we explain that? What drives the presence of these subclusters? It's kind of a chicken and egg question with the interferon signature. If it's driven by pDCs, why do they get enriched? And that's where we started to work with Jean-Charles, because it's very well known. There's high preponderance of females for autoimmune disease, and systemic sclerosis is one example. It's very clear also in Sjogren, in lupus, and others. And what's interesting about this is some of the genes that we know are critically involved in activating pDCs, TLR7 and also TLR8, are on the X chromosome. And it's very well known that if you have multiple copies of these genes, you can drive inflammation and lupus. So we had also shown that pDCs from patients express TLR8, while this is not the case in healthy controls. So this was another reason that we would be looking at TLR7 and TLR8. So the new hypothesis became whether the presence of these high IgG subclusters that we observe in patients, whether this is due to a defect in X chromosome inactivation at that locus in the pDCs of patients. So I have just a couple of slides on X inactivation, because just to refresh everyone's memory, it's kind of a complex system, but it's an essential one, because it's important that you have a dosage compensation between the 2 X chromosome present in females. And that was first hypothesized back in the 1960s by Dr. Marie Lyon, that the dosage compensation of sex chromosome genes has to be achieved by some form of X chromosome inactivation. So the mechanism now is much well defined. The role of X inactivation, so it's a long non-coding RNA that's basically being generated from the X inactivation center, and the GIST then start to spread and drops at very specific location into the X chromosomes. And by doing this, provoke various DNA and histone modification and basically repress transcription. So I'm not going to go too much detail either, just to say that this axis doesn't work by itself. It needs cofactors, and one of them is SPAN. And in absence of SPAN, it's almost impossible to have X inactivation. And there's all the complex, and then the maintenance is used through DNA methylation and others. But what's important to know is that although this is a well-established system, there's actually leakage into it. Many genes can escape X chromosome inactivation. Actually, more than 20% of all the human X genes can escape X inactivation and then be allelically expressed in the cells. But there's two types. There's the one that you call constitutive escapees. So basically, every cells, every time, these genes escape X inactivation. And then you have the facultative escapee. So they vary between cells and individuals, which creates this incredible mosaicism that almost every cell has its own combo of genes which are escaping and have two copies versus one, depending on the cell types in questions. So how can we look at this and what happens for the TLR7 and TLR8 situation? So this is again work that was done by Jean-Charles Guéry, and he had reported in a very interesting paper back in 2018 using cells from patient that has polymorphism on the TLR7. He could do kind of allele-specific single-cell RT-PCR in the female pDCs. And what he observed is that if you look into these cells, so these cells basically express only one of the allele. These cells express the other allele, and there's basically almost 9% of the cells that have two copies. So just in healthy cells, if you take out pDCs, there's about 10% of the cells that can escape where TLR7 can escape X chromosome inactivation. And the other approach where you can do this is doing by RNA fish. This is an actual reconstitution of staining when you see two copies of the TLR7 probes that get these emergent transcripts, and then the inactivated X is being identified by using as a second probe Xist. So you can see that here this is a B allelic expressing cells that has the TLR7 on the active X, but also presents into the inactive X. And so the control of this is important. They also reported that if you modulate a little bit expression of GIST, then you start to get autoimmunity with involvement of genes like 7, 8, or TASM. So what's the situation in patient then? So this is experiments just to describe it. Everything is done with primary cells, primary pDCs purified from patients from the blood. We had to make some adjustments because the technique is using quite a lot of cells, and also we only get, if you work with patients, you cannot get large number of cells to start with. So we had to merge patients and use them by batch. So we ran 4 different batches. This is the number of patients or subjects that were used and the number of nuclei that were studied. And we had basically a total of 25 patients, 1,500 nuclei for the patients, and 12 healthy donors, because we can get more cells to start with, and 2,000 nuclei. And then we ran RNA fish. And this is an example for TLR7. These cells is a monoallelically expressing TLR7, while here you can see the 2 dots with that cells, that pDCs being a B allelic. We found some TLR8, very few, some expressing, in particular more into the patients. And the B allelic was super rare. So I took a picture here, but having two copies of TLR8 was almost, I don't remember how many cells, but that was very, very, very rare. So, but now when we look at these 2,000 versus 1,500, and we look at how many cells actually are positive for these probes, you can clearly see an increase into the batches of the patient with SSC. And for TLR8, the healthy donors basically is very, very low, but you get these kind of higher numbers in patients with SSC. At that point, we say, okay, let's focus on cells which gives us a positive signals, and see how many of those are monoallelic versus biallelic. And that's how we found. So the healthy donors kind of confirm the initial work that Jean-Jacques Guéry had generated. You get 10-15% of the cells which have biallelic expression of TLR7. But look in the patients. It's like almost a third of the cells. A third of the cells have 2 copies of TLR7 as measured by RNA-FISH. I won't get into TLR8. It was a bit complex because there's not enough cells to really make a proper analysis. But I thought that was really, really stunning in terms of the observation. So we tried to link this with type 1 interferon. So first, we did this with a different technique. We did single-cell RT-PCR just to look at expression. And we saw not only more percentage of positive cells into the patient, but this is PCR, so you can actually now evaluate the level of expression. And we had about 1.8-fold more expression of TLR8, of TLR7 into the patient's pDCs as compared to healthy donors. So we know that interferon is important, and this is not completely unexpected. We know TLR7 can be induced by type 1 interferon. And indeed, if you take healthy cells, you give them interferon beta, and you run the same type of single cell, you can see an increase in the number, like expression of TLR7. But we also looked in the RNA-FISH expression, and similarly, we saw an increase in the number of nuclei that were positive, and also with an increase of cells that were expressing biallelic expression. So when we sent that paper, one of the reviewers asked us to do DNA FISH basically the risk would be that some of the probe that we use may not be as specific as we wish. And we're very happy that that question was asked because it led to something interesting. So the work was done by Julie Chaumer at Cochin in Paris. She's an expert of DNA FISH. And here what you see are human primary pDCs, which are being first on the left side probed by RNA-FISH, and then the cells are then used to conduct DNA FISH. And you have a lot of things in here. So in red, so this is a biallelic cells. In red is actually the TLR7 RNA-FISH probes. On here, it's a TLR7/8 DNA probe, just to make sure that we are on the same area. In blue here is the X-cyst, and X-cyst is going to stain for the inactivated X. So basically here, TLR7 is expressed where it's supposed to be, and here it's expressed on the inactivated X, as compared to monoallelic cells where it's only expressed on the XA, but it's not present on the inactivated X the way it's supposed to be, or the way it is in these monoallelic cells. So in the DNA FISH, so we confirm that the probes were read properly. So now in green is basically you paint the 2 chromosomes. So now you paint both chromosomes, irrespective of whether they are activated or inactivated, but that allow you to show that the TLR7 is basically close to the activated X. But as Julie was looking at this, she noticed that very often in biallelic cells, when she had TLR7 probe, it was kind of away from the actual painting of the X chromosome, as you can see into these cells. And so what she did, she actually quantified the distance between the center of the painting of the X chromosome and the actual probe staining for the TLR7. And what she observed is something like this, which you can basically also hear in quantification, is that when the TLR7 is transcribed on the activated X, it happens on the chromosome itself. But when it's actually being expressed on the inactivated, it looks like it's kind of moving away. It's forming like a little bit like of a loop outside of the cluster of the X chromosome to potentially allow expression. And she could quantify this. And as you can see, in total or in TLR7 negative, meaning in cells which are like in the DNA FISH, everything is inside. But once you look in the TLR positive on the activated X versus inactivated, it's only in the inactivated that is basically going away outside the chromosome, suggesting that in order to perform or for X chromosome inactivation the escape to occur, so you need to somehow have an impact on the actual structure of the inactivated X. So then we went back into our single cell cluster because we were interested to understand, but then what happened to all these regulators, exist, SPEN, and some of these other epigenetic regulators in the chromosome. And this is again just to show you basically exists into our data set. So it's expressed on every cells because we only have female patients and subjects. And basically what we observe is a reduction of expression of GIST into the pDCs of the patients. And this was not just exist. We saw reduction of SPEN, CBX4, while we saw an increase of ING4, and all this supporting basically an overall increase in acetylation, general overall increase in acetylation into the pDCs. And something similar had been observed in monocytes from patients. So suggesting again that there's a kind of a dysregulation of X inactivation into these patients. We looked for whether this was associated with type 1 interferon. So we went back into our clusters. So we had basically the ISG low clusters versus ISG high clusters, the clusters 0 and 2. But for this, there's no difference. So basically, it's reduced irrespectively of type 1 interferon. While SPEN, there's a bit of an inverse correlation between the presence of interferon and the presence of SPEN. Same with CBX4. And we were very fortunate when the paper came out that Monsera Angera and Nikki Jiwaka wrote this nice commentary that they basically summarized the finding that we had is that this kind of female bias disease associated with coding signature could be associated with a defect into X chromosome activation into the pDCs. All right. So then I'm going to switch gears slightly and present a couple, not that many slides, just a few slides on basically what happened to the pDCs when they reach the skin. Because this is something that kind of bothered me for some time is we know pDCs are not present into the skin. And in patients, in lupus, in scleroderma, if you create injury, if you do tape stripping and others, pDCs rush to the skin and make type 1 interferon. And we know that the skin is a very different environment than the blood. The stiffness is much higher, even in healthy skin as compared to the blood. It gets much stronger in fibrotic skin. It can reach 50 kilopascal and probably even more than that, depending on the type of skin. So we use a model using hydrogels, which are polyacrylamide, which are cross-linked, and where you can basically seed the pDCs on top, and they coated with type 1 collagen, and then you can just trigger basically mechanosensing into the pDCs. And I have to say the hypothesis that I had initially is that this was probably going to be sensed as a danger signals, and that as you increase stiffness, the cells would then respond and produce more type 1 interferon. So as you can see in here, I was absolutely wrong. If you take primary pDCs from healthy donors and you stimulate them with a CpG, with a classic dose of CpG, and you quantify type 1 interferon, as you increase the stiffness, their ability to make type 1 interferon is actually going down. Same as if you use the TLR7 agonist. So obviously we had two questions. What's the mechanism leading to this? But then why do we see chronic interferon expression in the skin of these patients? Makes no sense because it's even more fibrotic. So first question, we excluded the classic YAP-TAZ pathway. Is basically YAP is just not there. The TEDs, which are the transcription factors needed for expression for the modulating inducing the response are not expressed. Not only we looked at primary pieces from the blood, but when we reanalyzed the sample from that same studies in patients, they basically had the same findings. If you look in the blood or in the skin, in both cases, YAP is just not expressed and the TEDs are not expressed. So we know it's not associated with the YAP-TAZ. We run a quick RNA-seq comparing cells culture and condition at 50 kilopascal versus 0.2 kilopascal. And what really came out is NRF2. It's not alone. There's a lot of stress pathways coming out. Stress genes like ATF4, XBP1. I mean, clearly there's genes associated with stress induction that seems to be induced by the high stiffness. And this was associated with reduction of all the interferon genes, which made sense because this was the observation. So basically, you induce NRF2, and that leads to reduction of the type 1 response. We confirm this. We actually do see an increase of NRF2 in cells culture at 50 kilopascal, or if you use Ki-696, which is an inducer of NRF2 activation. And if you take cells and you indeed use that molecule to on top of the CpG, you reduce type 1 interferon response. This is another drug. And importantly, if you put the cells at 50 kilopascal, they cannot make as much interferon in response to CpG. But if you block NRF2, you can rescue, and then these cells then now can make type 1 interferon. Once again, question became then, what happened? Why are the cells from patients still making interferon in the skin? And we went back to reanalyze again these data sets. And what we found was quite interesting is that now if you compare the pDCs from patients, skin versus blood, so this is all patients, skin versus blood. We cannot do this in healthy donors. There's no pDCs in the skin. So you do see NRF2. So you see that NRF2 is being induced into the skin when pDCs reach the skin of these patients. But so is RF7. So now it's kind of uncoupling of these two pathways. NRF2 is still there, but they can still make type 1 interferon. So how does that work? We actually even looked at the very NRF2 high or NRF2 low expressing cells, and it just has much of an interferon response in the skin of the patients. So we think this has a lot to do with CXCR4. CXCR4 is actually present into the skin. We had observed that, at least in the mice, it's very clear in fibrotic skin. We know that if you take pDCs and you hit them with CpGs, kind of a suboptimal dose, and you add CXCR4, it really largely increases the expression of type 1 interferon. This has been confirmed by many other groups like Roberto Lande and others. So we did the experiment. We put the cells, stimulate them with CpG, either in normal condition or at 50 kilopascal, and then you see a reduction of the interferon response. We did the same with Ki-696, see reduction of interferon response. But then if you supplement with CXCR4, you completely overcome that inhibition, suggesting that a lot of these regulations are kind of solos. And then the presence of inflammatory mediators like CXCR4, and I would even argue potentially if you start some cells which have different profile to start with, that can drive an actual type 1 interferon response. And we confirmed that into the mouse. When we use the tape stripping models, you look at this is in pDCs, you get induction of MX1 or ISGs and others. If you give them an RF2, it goes down, but that is completely blocked by the presence of CXCR4. So in conclusion, this is the model that we have. We think that following tissue injury, inflammation, and others, pDCs are irremediably attracted to the skin, where they're going to be produced type 1 interferon, activating cells in proximity, like macrophages, for example. This is going to change the environment in the skin with activation of fibroblasts. To fibroblasts, that's going to increase stiffness, and that is actually part of the normal wound healing process. And the increased stiffness that you see in the wound and in the scarring is basically a way to also terminate the response by pDC and being sure that there's no longer presence of an interferon signature into that skin. They came in, they helped, they contributed to the wound healing, but now the response is terminated through induction of stress pathway and genes like NRF2. In contrast, in scleroderma, you have other mediators which are present. CXCR4, type 1 interferon, but also the cells to start with are different because they come in, more of them have this biallelic expression, they have more interferon response, and it cannot be as inhibited as would be normal cells, leading to the persistent expression of the type 1 interferon and persistent fibrosis. So this was a very interesting review that was just came out in Nature Review Rheumatology from it's called An International Perspective on the Future of Systemic Sclerosis Research. And their main conclusion was the pathogenesis is very complex, interplay between environmental factors, exposures, host genetics, target organ injury, immune activation, and fibrosis. And this is basically the summary table that they had. And I just thought I would add a little bit of complexity by adding the pDCs that somehow they did not include into their review through the presence of type 1 interferon and other factors like CXCR4 that can promote this very profibrotic phenotype. All right, so just to thank the people that did the work, all the work that I've shown have mostly been done by Vidyanathan Chaudhary and Yangdu, two postdocs in my lab. Yang did the work on the X chromosome inactivation and single cell, and Vidyanathan all the mechanosensing work. Dominique had done a lot of the initial work characterizing pDCs in patients. We had contribution by Max and Bikash in the Ivashkiv lab, looking at the pDC macrophage interactions. There's nothing I could do in this place without my collaboration with the clinicians in the Scleroderma and Vascularity Center, in particular Kim Lakin, Jessica Gordon, and Rose Piera. We have project also in lupus. This is a very close collaboration with the Jean-Charles Guéry in Toulouse and also Julie Chaumet at the Cochin Institute in Paris, but also with Marie-Élise Truchetet in Bordeaux, which very help us recruit, characterize some of the patients that we use for that studies. And although I have to say, I don't have any slots for postdocs right now, I still put that slide anyway, because I know people here are looking for postdocs, especially if funding becomes better. So New York is a nice place. So that's what I think. All right, thank you very much.
All right, thank you, Frank, for really interesting and thought-provoking seminar. Let's see if we have any questions.
Maybe I should stop sharing.
Yes, you can do that. So I'm going to read a couple of questions. I don't know if you see them as well, but the first one is from an anonymous attendee. Thank you for your fascinating presentation. Do you know at which stage of development escape from X inactivation occurs for TLR7 in pDCs? Does it occur in common lymphoid progenitors? What does that mean for T and B cells?
So well, the straight answer is we don't really know. But what has been shown, at least in healthy donors, is that the escape is very across all cell types. So what you see in pDC is very likely open also in other cell types like monocytes, and T cells, and B cells. And actually, Jean-Charles had a follow-up study on this looking in monocytes at the escape of TLR8. So it's not uniquely, I should have said that. So it's a very good question. It's not uniquely restricted to pDCs. It's important for pDCs because there's the type 1 interferon associations, but it's not restricted. So whether it occurs very early, I think that's the common thinking, that it comes in some of the progenitors, and that's why it's equally distributed. But I don't think anyone has actually done like tracing of this to see whether that's the case.
So I'm going to insert my own little question here to follow up on that. Do you know if type 1 interferons can affect X chromosome inactivation, maybe negatively?
So yes and no, because it does influence expression of SPEN, but not of XIST. So if you we haven't done this, but it's been reported in other cell types. If you culture cell with type 1 interferon, it does not reduce XIST.
Okay.
So it's not causing reduction of XIST. So it's kind of a chicken and egg. It's hard to really know what comes first.
Okay.
Yeah.
So the next question is from Natalia Giltier, who says, super interesting, thank you. First question, is SLE pDC's activation driven by immune complexes? Is there an antibody immune complex component in the driving tissue inflammation in scleroderma?
Yeah, I mean, this is kind of a likely scenario. I mean, this, especially when you know that some of the subtype of patients with systemic sclerosis are also associated with different type of autoantibody, like topoisomerase, for example. So it's very likely that, similarly to what's been shown into the in lupus, that would be the same case in scleroderma. We haven't actually done this experiment properly, but that would be the expectations that you would see the same.
And then Natalia's second question is, in in addition to X chromosome inactivation escape, increased TLR7 expression can be driven by SNPs in the TLR gene. Have you looked if scleroderma patients have a higher percentage of the TLR7 risk allele?
So we have not, but that's a good point because you need to understand what drive this, right? We see we haven't been able to look at whether the escape was really in these ISG high subclusters, because you would have to somehow, we assume that's the case because we see these high subclusters, we see escape, and then we see these kind of changes in expression of some of the histone modifiers, if you want. But we actually haven't looked precisely whether that's the case. And risk allele of TLR7, maybe that's what starts the whole process. And then you have this kind of slow enrichment of this population from 10 to 12 to 15 to 18 to 20% to 25% until kind of break the things, break tolerance, and then you start inducing disease. So this risk allele could really play a role in that context.
So could the risk allele help in escape from
Yes.
inactivation?
Because as soon as you give, increased signaling of TLR7, even like, this whole TLR network has been very evolutionary tested. I mean, this is to trigger immune responses.
Mm-hmm.
So very small changes can have large consequences. As you probably all know, if you just take mice, you put 2 copies of TLR7 in normal mice, they start developing some kind of autoimmune phenotype. So it's very subtle differences. So yeah, maybe a risk allele might just be enough to get the whole cascade started.
Yeah. We have a question from Mark Wenner. Fascinating, thank you. Male scleroderma is less frequent but more severe often. What is known about exist expression in X chromosomes of men with and without autoimmune disease?
So X is not expressed in men. I know this is recorded, so I should pay attention, but I don't think anyone has ever been able to found exist expression in male cells.
Mm.
So this cannot be the driver of what's going on in 2 males. I mean, I know there's this very fascinating work by Howard Chang when he has actually expressed exist into males, and by making a modification so it would lose its repressing function, but would still be present, and then it can drive autoantibody response and activation of TLR7. But like in normal male cells, I don't think exist can be expressed.
So how does the exist locus on the X chromosome know if there is another X chromosome or not?
That's a question for Edith Hurd or someone else, but yeah. But the X chromosome activation is very different in between species. There are species where it's basically locked in at the embryogenic level. It's only going to be the father's X that's been inactivated. In some, like I think in mice, it changes at a certain level of embryogenesis, while in human, it's stochastic all the time. Mm. So there's different models in terms of how that works.
Oh, interesting. Okay, so the next question is NRF2. Can you tell us what you think about the overexpression you observed, said to have a role in oxidative stress? Is there a way to connect the vasculopathy of scleroderma?
Well, yeah, I mean, there's been people looking at NRF2 and its role in vasculopathy in doing fibrosis and in patients. So I think that's a very valid point. We haven't looked really in detail because we only see the induction of NRF2 as kind of a stress response. And this was kind of a nice follow-up on the work we had been doing a few years ago on ER stress response in pDCs. So pDCs are very dependent on like an optimal metabolic status to make type 1 interferon. As soon as you divert a little bit away from the TCA cycle and then you reduce OXPHOS, they don't make interferon anymore. So NRF2 is one of these ways, but we have same data if we put cells in the hypoxic environments. So it's not sure that the vasculopathy is being driven by pDCs per se. So that's why I'm a little bit reluctant to make that link at this point.
Okay. So the next question is, in general, does TLR7 escape of X inactivation immune cells change with age or over time?
Yes, the answer is yes, because when we first did the experiment, we had this incredible data, but then because we are doing with the samples that we had, and we realized like some of the female healthy donors were much younger than the patients. And then once we started to recruit healthy donors of age match, then the difference was not as pronounced. So clearly, yes, this goes worse with age. As you age, this mechanism is being lost.
So I'm going to ask you one question of my own. In the case where you had a biallelic expression of TLR7 and it was physically removed from the bulk of the X chromosome, do you know if
Yes.
it was then closer to the nucleolus or somehow placed close to where splice factors would be more abundant? So, sort of a more favorable region for transcription and maybe processing of the nascent transcript.
That's a good point. I don't know. We haven't thought about that. It's possible. I mean, to me, there was kind of my naive view was that, you see TLR8 expression into cells of patients. And I thought, well, maybe because they escape and 7 and 8 are so close one from another, that somehow you lose expression of TLR8 mostly from the inactivated X. But I haven't thought the fact that you get out of with this loop, somehow it makes you in a more favorable transcriptional wise environment. Yeah, it's a good point. We haven't thought about that.
Yeah, well, we can discuss it more in a moment on a
Yeah.
different Zoom link. Are there any questions more in the audience? I don't have any more submitted at this point. If not, I think we will then thank you very much for a really interesting seminar, and I will talk to you in a moment.
Thanks
Okay,
all.
thanks.